Good evening, everyone. We'll wait for a few more minutes to let others join and then we can start. Hello, good evening. We are just waiting for a few minutes to let others join and then we'll start today's session. I guess we can get started. Let me just share my screen. Yeah, I hope you're able to see this. Okay, good evening everyone, and welcome back to week 9 of NPTEL online course Drug Delivery Principles and Engineering. Let's start with some of the points that have been discussed in our previous sessions, as well as a recap of previous session points, as well as some key points that were discussed in week 9 lectures in the course. So, until now, we have talked a lot about drug delivery, we have talked about tissue engineering principles, and in the last few sessions, we have talked about different routes of drug delivery. So, we started off with the oral route. So, in this, we saw that oral route is the most patient compliant route of administration for drugs because it's really convenient for people to take pills. These could be the drugs could be formulated in the form of tablets or capsules or a syrup, and in that sense it becomes very patient compliant, but it also has its own disadvantages. Yeah, it also has its own disadvantages in the sense that any drug that is given orally suffers from the first pass metabolism. What does that mean? It means that the drug gets metabolized even before it reaches the site of action, so we lose a lot of amount of drug in this process. As a result, to make it therapeutic, you have to deliver a lot of amount of drug, and this could lead to toxicity effects also, so you have to balance the dose in that way. And another drawback of oral route was most of the drug gets degraded in the gastrointestinal tract itself, wherein before it gets absorbed for the systemic circulation. So these were some advantages and disadvantages of the oral route. So if we want to avoid the first pass effect, then there are various different routes of drug administration, right? So these could be your parenteral routes, meaning it could be subcutaneous, intramuscular, wherein you inject something. So you have your skin layer, the dermal layer first, then you have your subcutaneous layer, and then you have your intramuscular layer. So advantage of these routes, it eliminates the first pass metabolism, but certain disadvantages is there's a limit to how much amount of volume you can put in these with these administration routes. And also, since these are injections, you have to only trained people can administer these. It's a little difficult for the patient to themselves administer it, so they have to get trained for it. And also, since these are injections, we are injecting something in the body, then it could cause irritation at the site of injection. It could lead to inflammation as well. Then we saw transdermal. So in transdermal, this is also a very patient compliant route of administration because you just put a patch. Yeah, you can deliver the drugs in the form that you can formulate it such as it's in a patch, and you can put that patch on the skin, and so you have micro needles to do this. So in that sense it becomes patient compliant because then people don't have to, they can self administer it, you don't have to get trained for this. The disadvantages of transdermal route is we have a very thick layer of lipids as well as dead cells on our skin, so it becomes a little difficult for the drugs to penetrate through this layer. But we also saw different ways of enhancing drug delivery through this transdermal route, right? We saw there are certain chemicals which can enhance the permeation of the drug through the skin, for example ethanol or DMSO. So these are about chemical enhancers. Then we also saw certain things wherein you can apply an electric current to the skin, not anything to harm the patient, but enough to destabilize the dead cell layer so that the drug can easily pass through. So we saw that as well in the previous lectures. And then we also talked about intra-articular. This is a very specific method to deliver drugs to the knee joints in case of osteoarthritis, for example. So that was about intra-articular. And then we also saw an intravenous route. So this we people must be more familiar with because we have seen people in hospitals getting IV solutions. They have saline passing through, or they have drugs passing through IV route. So advantage of this route is first of all it's instantaneous, and it has the highest amount of bioavailability because anything that gets delivered through intravenous route immediately reaches the blood and through the blood to all the parts of the body because of the systemic circulation. So in that sense intravenous is very advantageous. If you want an immediate effect of the drug, intravenous is the best route to go ahead with. Disadvantages is again it needs trained personnel to administer drugs through intravenous route, and also it could be invasive. It is invasive because it's an injection, and since everything gets delivered to the blood and then through the blood to the entire body, there are chances that if your formulation is not very sterile, then it could lead to infection and certain immunological reactions as well. So this is what we saw in the recap of the previous lecture. In week 9 lectures, then we start discussing a little bit more about the immunological aspect of drug delivery and implant implantation, and how the body responds to anything, a drug being delivered, or a biomaterial being put into the body, or an implant being put into the body. So it starts with this topic of differentiating the immunities that a human has. So we have innate immunity as well as adaptive immunity. So innate immunity is very non-specific and immediate. So you must have seen this. So the way, let's say I get a bacterial or a viral infection, my non-specific and the innate immunity is the first one that's going to launch an attack against this particular pathogen, be it a pathogen or be it a foreign material. So the immediate the immunity, the branch of immunity that takes an immediate action against this pathogen is the innate immunity, versus we have the adaptive immunity. So this comes in a little later after the infection has happened. It's highly specific, and so it has a late onset, it starts a little later, and then also has a longer duration. So that's where your B and T cells come into play for the adaptive immunity. The innate immunity is handled by your neutrophils and your complement systems, which are very non-specific. Anything that they think is a foreign or a pathogen, they'll start attacking that. Versus if you have adaptive immunity in play, these cells are very, very specific to a specific antigen, and so they provide immunity for a longer duration. So that's what was discussed in these lectures. And then you have inflammation, right? Inflammation forms a very important point when you're trying to put an implant, or let's say put a biomaterial. So this is a cascade of events. So what happens is, let's say I'm trying to implant something into a patient's body, and in the process of this implantation, there is a possibility that some of the blood vessels, the tiniest blood vessels around this particular area, might get ruptured. So there is an injury in the patient's body, or the body will see this implant as a foreign entity. So what happens is, we have a lot of macrophages, we have a lot of dendritic cells that are surveilling the system, right? They're just moving around, they are there in the vicinity of this implant. So they'll think that this is a foreign material, or they get triggered because of the injury that has happened while implanting this particular biomaterial. They then get activated, and they start secreting cytokines. So cytokines are very small proteins, and they are signals to other cells that some injury has happened, or there is some infection, or there is some foreign body in the human body. So because of these cytokines, then certain processes happen in this inflammation. So first of all, there is vasodilation. What does that mean? We'll also discuss about this in detail in the upcoming questions. Vasodilation is nothing but the blood vessels start dilating a little, so that more and more number of immune cells can come to that area. There's an influx of leukocytes, meaning the immune cells start coming to this area because of this cytokine signaling that has been given by the immune cells that get activated because of the injury, and eventually it leads to healing. So in this process, if the inflammation is not resolved in the body, it could lead to fibrosis. So that could happen on the implant surface itself, and this could then hamper the drug delivery the drugs that were supposed to be released from this implant. So it could hamper that, and eventually it could lead to device failure as well. Since there's a constant immune attack being launched because of this implant, then the final solution to avoid this problem would be to remove that implant. So you can't keep it in the patient's body if the inflammation is not resolved. That was one point that was discussed. And then we start looking into details of these immune system. For example, a complement system. So as I said, it's part of the innate immunity. It's non-specific. The job of complement system is to clear microorganisms, and there are different pathways. One of it is alternative pathway, which is the most relevant to biomaterials. So whenever you put a biomaterial in the patient's body, your complement system is going to be triggered. And there are ways to avoid this. There are ways to minimize this activation of complement system in order to not let the immune system of the patient's body to hamper this biomaterial. So we'll discuss about complement system as well. It's one of the questions in the upcoming assignment. So what happens when you implant a material inside a patient's body? First of all, so blood flows all over the body, right? So wherever you're going to put an implant, blood is going to be in contact with the implant, and so as I said, this complement system gets activated. So complement system is nothing but it's made up of different proteins, which are these proteins usually are in an inactive form in the plasma, in the blood. So whenever they come in contact with a pathogen, or they come in contact with a biomaterial that's not part of the body, they will get activated, and then they further start a cascade. It increases the immune response. They start a blood clotting cascade as well, and as I said, this might lead to device failure because of fibrosis. And there are ways to reduce complement activation. You can modify the surface in such a way that these proteins that I said, which form the complement system, you can reduce the interaction of these proteins with your biometal surface. So in that case, there are ways to minimize this complement system activation. Yeah, and the last point in these week 9 lectures was going further into adaptive immunity. So what cells make part of adaptive immunity? These are your dendritic cells and macrophages, and also your T and B cells. So dendritic cells and macrophages are very phagocytic in nature. Their job is to look for any microorganism or any foreign material that is present in the body. They phagocytosis, meaning they'll just eat up this particle, and they'll chop down the particles. The mechanism present in these macrophages or dendritic cells, they will completely cleave this particle, and they'll present a small part of this particle on their surface. So this is known as antigen presentation. Now your B and T cells are very specific to in identifying these antigens. So they will identify these antigens, and your T cells might directly kill the particular pathogen, or in case of your B cells might can start producing antibodies against this antigen, and in that process they'll try clearing of the infection. So yes, this was in terms of how the adaptive immunity functions in the human system. So this is more in context with the vaccines, so that we'll be discussing in the next session. So that's a recap of most of the points that were discussed in this week's lecture. So if you people have no doubts, then we'll move on towards the assignment questions. Okay, let's look at the questions then. The first question is, a non-compatible implant was used to deliver drugs in vivo. Which of the following activities will be triggered? So we have to choose two correct options. So the question is, we already know that the implant is going to be non-compatible, but it was used to deliver drugs in vivo in a patient's body. So which of these following activities will be triggered because of it? So take your time in reading through the options and let me know if you have the answer. If not, we'll discuss. So you can unmute yourself and let me know the answer, or you can type it out in the chat box as well. That also works with me. But yeah, let me know if you know the answer. If not, we'll discuss. Yeah, I guess somebody is trying to answer 2 and 4. Thank you for responding. So the second option is, there may be higher chance of deposition of fibrotic tissue onto it, and the intrinsic clotting pathway could be activated. Okay, thanks for answering. All right, let's look at the answer then. The correct answer is whoever answered in the chat box, you're correct. The correct answer is option 2 and 4. So that's what happens when a non-compatible implant is put to deliver drugs in vivo, right? So first of all, what could happen? There could be fibrosis happening. A fibrous capsule forms over the drug delivery system, over the implant, and then this could lead to diffusion limitations, right? Now that so you have already formulated your drug delivery system to have a certain release rate of your drug. Now if you have a layer on top of it, it is going to create problems, because then the released drug could maybe is not able to diffuse into the body because of this fibrous capsule forming over it. There could be limited exchange of fluid. Maybe the way you formulated your implant could be you needed water to diffuse into it. For example, in hydrogel systems, you need water to diffuse into it for the drug delivery to happen. Maybe that could be limited. In that case, not enough drug would be released. There could be slower rate of hydrolysis and degradation. It could lead to slower rate of drug release, and this could then be dangerous to patient, right? You never expected fibrosis to happen, and then you calculated all the dose, you calculated the release kinetics, you know how much amount should be getting delivered in particular time, and then you have given it to the patient. Now if that rate is not being maintained, if not enough amount of drug is being generated, that could be dangerous to the patient. So your first correct answer for this question should be there may be a higher chance of deposition of fibrotic tissue onto it, and then the intrinsic clotting pathway could be activated. So the intrinsic clotting pathway is nothing but that gets triggered because of the surface of the biomaterial. So the other options are incorrect for this. The correct answer is option 2 and 4. I hope this is making sense, and if there are no doubts with this particular explanation, then I'll move on towards the next question. Okay, I'll move on towards the next question then. Next is, which of the following are the professional antigen presenting cells of the innate immune system? And we have to again choose two correct options. So we just discussed about this in the introductory slide. So take your time in going through the options and let me know if you know the answer. Okay, we have a response option 2 and 3, macrophages and dendritic cells. Okay, thanks for responding. If anybody else would like to give it a try, please go ahead. Oh, somebody else is also answering 2 and 3. Thank you for answering. Let's look at the answer. Yeah, both of you are correct. The antigen presenting cells of the innate immune system are nothing but macrophages and dendritic cells. So this you will also come across when if you're going through the lectures from this course. So your dendritic cells and macrophages, they are very phagocytic. Their job is to present antigens to B and T cells, and they're always present in the tissue and they're surveilling the surroundings. So whenever there's infection, so that's how infection gets controlled in our body, because these cells are always surveilling the tissues. Whenever they come across a pathogen, they'll start engulfing that pathogen, chop it up, and then present the antigen on their surface. So they present it to the T cells. So T cells are of different types. They could be your helper T cells, they could be your cytotoxic T cells, or they could be your regulatory cells. All of these are involved in controlling the infection. They start killing the cell. So it could be a pathogen, or it could also be a intrinsic cell belonging to a human body which has gone mutant. So they also play an important role in controlling cancerous cells. And your B cells then are responsible for generating antibodies against the pathogen. So your dendritic cells and macrophages are the professional antigen presenting cells of the innate immune system. I hope this is clear. We'll then move on towards the next question. Next is, which of the following are uptake mechanisms at the cellular level? Again, we have to choose two correct options. So what are the mechanisms which are used by the cells to uptake things at a cellular level? Okay, people are saying second and the last option, clathrin mediated endocytosis and pinocytosis. Okay, you're correct, whoever is saying option 2 and 8. So these are some of the mechanisms by which cells can uptake things. So let's just go through the slide. So what is phagocytosis? This I was saying, right? Your dendritic cells and your macrophages are very phagocytic in nature. So what they do is they have their plasma membrane. If they sense that there is a foreign particle in the vicinity, they'll just close over this plasma membrane, and in that process they'll just engulf the particle. So that is one method, phagocytosis. What does pinocytosis mean? It is also known as cell drinking. So if there's an extracellular fluid here, the cells will just start taking up this fluid in vesicles. So that's pinocytosis. And then there is receptor mediated endocytosis. So if you see the where my pointer is at, these red markings, right? So these could be your clathrin. It's a protein that is intracellular to the cell membrane. So with the help of, so you first have receptors on the surface of the cell. So these receptors are very specific to their ligands. So once a ligand binds to its receptor, these clathrin proteins then get activated and they will pinch off the cell membrane, and in that process they'll uptake it helps the cell to take these particles inside the in the cytoplasm of the cell. So some of the mechanisms at cellular level are clathrin mediated endocytosis and your pinocytosis, option 2 and 8. So thanks for responding. for people who are typing it out in the chat box. If there are any doubts with this, I can wait for a few minutes. If not, then we'll move on towards the next question. Thank you for answering. Okay, no doubts. Then let's look at the next question. Next is, which of the following proteins is not related to the complement pathway? Again, we have to choose two correct options. Please read the question carefully. We have to choose proteins which are not a part of complement pathway. Okay, people have already started answering last two options, P53 and P10. Anybody else would like to give it a try? Okay, let's look at the answer. All of you are correct, whoever is answering option 7 and 8. So the rest of the proteins, so these are the proteins that are involved in the complement system, right? So as I said, they get triggered when there is a membrane here. So all of these that are listed out in the option C3, C5, C1, they are directly involved in the complement pathway. Properdine, factor D and factor H, they are involved in, so properdine is a protein that is involved in stabilizing these particular enzymes C3 and C5. Factor H is involved in the control of controlling the activity of these enzymes, and so on. So the only option that doesn't fit into complement pathways is P53 and P10. I hope that's pretty straightforward. If there are no doubts, we'll move on towards the fifth question then. Okay. So the fifth question is, which of the following cells are the first to reach the site of inflammation? This should also be pretty easy to answer. Please let me know if you are you already answering. Okay, somebody's answering option 2, neutrophils. Thank you for responding. Anybody else? Okay, let's look at the answer. You're perfectly correct. Neutrophils are the first cells that reach the site of inflammation. Not much explanation with this, I guess. It's pretty straightforward. Let's move on towards the next question then. Next is, what are the implications of a foreign body response to an implant? Again, we have to choose two correct options. For people who have joined a little late, we have already started with the assignment, and if you people have missed some of the initial questions and want me to go over them again, then we can do so at the end of the session. Do let me know if you people need it. Otherwise, we'll just follow up the rest of the questions of the assignment. Okay, people have started answering. Most of them are saying option 1 and 2. Thank you for responding. Anybody else? Right, let's look at the answer. Yeah, all of you are correct. Whoever answered option 1 and 2. So, we just talked about this, right? What are the implications of foreign body response to an implant? So, if you have designed an implant which is to deliver drugs at a constant rate, or some release kinetics which you have already formulated, and if that generates a foreign body response in the patient's body, then all of these could affect the drug delivery. First of all, there could be fibrous capsule formation, there could be a layer which covers your implant, and then this could lead to problems in drug delivery. First of all, it can have diffusion limitations. Your drug will not be able to diffuse from this implant to the rest of the body, which was the whole purpose of putting this implant. It could also lead to limited exchange of fluid. Water will not be able to flow into this device at the rate that you had calculated upon. Again, it could disturb your release kinetics that you had decided, and it could lead to slower rate of hydrolysis and degradation, which would then ultimately change the rate of drug release. And in case it's not as what we were expecting when we actually put the implant, then that could be a little dangerous to the patient as well. So these are the implications. So if you just follow up with the options here, first is limited diffusion. Yes, that could happen. That is one implication of foreign body response. Limited exchange of fluid. Yes. Increase in microvasculature, increased coating of lipids, increased exposure to dietary elements, decreased coating of proteins doesn't at all matter in context of foreign body response to an implant. So the correct answer is option 1 and 2. Clear? Okay, then let's move on towards the next question. Next is, which arm of the complement system is first involved in the body's interaction with the biomaterials? So we just discussed in the introductory slide, right? So there are different ways through which complement system can get activated. So one out of these is most relevant in case of biomaterials. So if you people know which one, please let me know. If not, we'll then discuss. Okay, people have started answering option C, alternative pathway. Thank you for responding. Anybody else would like to give it a try? Right. Thank you all of you for answering. So the correct answer is option 3, alternative pathway. So as I was saying that the complement system can be activated in various different pathways, the classical, the alternative and the lectin mediated. So the most relevant for biomaterials is your alternative pathway. The classical pathway and the lectin mediated pathway is more in terms of your pathogens or your infections, microorganisms that can cause diseases. So the complement system also takes care of that. But in case of biomaterials, we just saw in the previous question, right, there are the proteins of these forming these complement system, they can get attracted, they can get attached to the biomaterial, and they can view the biomaterial as a foreign material, and then they can get activated. So the most relevant complement system pathway for biomaterials is the alternative pathway. I hope this is clear. Let's look at the next question then. Next is we wish to generate an osteoconductive implant by coating it with fibronectin. For this purpose, an aqueous solution of fibronectin was prepared. Assuming the surface of the implant is positively charged, how should we change the pH of this solution so that a large amount of fibronectin is attached to the surface? And we have been given the PI value of fibronectin, which is 5.75. So we have to adjust the pH in such a way that maximum amount of fibronectin gets attached to this particular implant. So if you people know the answer, do let me know. Okay, people have already started answering. Option 1, make pH lesser than 5.75. Thanks for responding. Anybody else would like to give it a try? Right, so most of you are answering as option 1, make pH less than 5.75. The correct answer is actually make pH more than 5.75 at 37 degrees Celsius. Option 2, let's look why. So let's understand the question first. So you have an implant, and you for sure know that the implant has a positively charged surface, and we have to Then, so the purpose is coating fibronectin over this positively charged surface. So the most logical thing that we know for sure is the fibronectin has to be negatively charged, so that maximum amount of this negatively charged fibronectin then gets coated over the positively charged surface. Now we know that pi of fibronectin is 5.75. So what would the pH of the solution be so that fibronectin is completely is negatively charged? So for fibronectin to be negatively charged, the pH has to be more than 5.75. If it is less than 5.75, it will create a positive charge in fibronectin. So we don't want that. The positive charge of fibronectin and the positive charge of the surface will then have repulsion, and most of your fibronectin will not get coated over the surface. So you have to have negatively charged fibronectin. For us to do that, you have to keep the pH above 5.75. And why 37 degrees Celsius? Because it's your body temperature. So better to keep everything at 37 degrees Celsius while you're doing this, so that it mimics the body temperature. So the correct answer is you keep your pH above 5.75 at 37 degrees Celsius. Is this clear? I hope there are no doubts with this. If you people want me to go over this explanation again, I can do so. If not, then if it's sufficient and you people have understood this question, we can move on. Okay, clear? Let's look at the next question then. Next is, which of the following is not a physiological event occurring during inflammation? Again, we have to choose two correct options. So we just saw what happens in inflammation, and if you people have read about this, if you people have gone through the lectures for this week, it should be pretty easy to answer. Just give it a try, read through the options. I'll wait for a few minutes. If not, then we'll of course discuss. Anybody else? Okay, let's look at the answer. So the correct answers are reduction in temperature at the site of inflammation and migration of immune cells away from the site of inflammation. So please read the question carefully. We are supposed to list out events that are not occurring during inflammation. So if you just follow the options, vasodilation happens because we just saw, right? So because of the inflammation, whenever macrophages get activated, the immune cells will get activated because of some injury or some infection. They will start releasing cytokines, which would then increase the diameter of the capillaries nearby the inflammation area. So this is done so that more and more immune cells can come and infiltrate into the tissue. It increases the capillary permeability. I'm just going through the options. So that also happens because your immune cells are flowing in the bloodstream, right? They have to reach the tissue. So for them to do that, there is increase in capillary permeability. More and more cells can squeeze through the capillaries into the tissues where the inflammation is occurring. There is an influx of protein rich fluid and cells at the site of inflammation. Yes, your immune cells are being attracted towards the site of inflammation, and then they start infiltrating into the tissue. Swelling occurs because of this, because now your blood vessels are getting dilated a little. More and more amount of fluid is incoming in that tissue, so that's why you see swelling in inflammation. Reduction in temperature at site of inflammation is incorrect. So you must have seen, right, whenever you get an injury or whenever you just hit your hand against a table or something, you'll see that it starts swelling a little, and that particular area is a little warm. So temperature actually increases at site of inflammation, so this option is incorrect. There is pain at the site of inflammation because of all of these immune cell infiltration, and these the area starts swelling, it starts pushing on the nerve cells a little, so that's why you experience pain. And then you have an influx of leukocytes and phagocytic cells into the tissue. Yes, migration of immune cells away from the site of inflammation is incorrect. Immune cells actually move towards the site of inflammation. So which of the following is not a physiological event occurring during inflammation? Reduction in temperature and migration of immune cells away from the site inflammation. This is what doesn't occur during inflammation. Is this correct? You can read more details about it in this slide as well. Oh, people have answered 5 and 8. Sorry, I didn't look at it before, but 5 and 8, yeah, you were correct. Clear with this question? We'll move on towards the next question then. Next is, which of the following is not an action of the complement system? Again, we have to choose 2 correct options. Yeah, so the last question is, which of the following is not an action of the complement system? I'll wait for a few minutes for you people to go through the options. If anybody knows the answer, please let me know. No responses. Okay, somebody saying 2 and 6. Okay, 1, 2, 3. Okay, thanks for responding. Anybody else? Alright, let's look at the answer. So yeah, you're correct. Option 2 and 6 are the correct options for this particular question. So we have to again choose what is not an action of complement system. So you'll be able to answer it if you know what complement system actually does. So there are a number of functions that complement systems carry out. Most prominent of these are, first of all, it triggers inflammation. So we just saw, right, what happens when inflammation occurs. More and more immune cells come at the site of inflammation. So complement systems are responsible for triggering this inflammation. They chemotactically attract phagocytes to the infection or the implantation site. So they have this ability of attracting your macrophages, your dendritic cells, to the injury. They also promote attachment of antigens to phagocytes. This is also known as opsonization, so that your phagocytes are able to target these antigens in a much more prominent way. They cause lysis of gram-negative bacteria and human cells which are displaying foreign epitopes, and they remove harmful immune complexes from the body so that it doesn't further lead to increased immune attacks. So somebody also has to remove these immune complexes. So that job is done by your complement system. So if you just see the options, trigger inflammation, that is done by complement system. It repels infiltrating leukocytes from the site of infection or inflammation, that is incorrect. It actually attracts these leukocytes. Attracts phagocytes by moonlighting as chemoattractants, Correct option. Fourth option is again a repeat of the third option. That happens. Promote the attachment of antigens to phagocytes. Yes, that is, we just saw it. It's opsonization. It protects foreign objects or organism for degradation. That is incorrect. It actually leads to foreign object degradation. Causes lysis of gram-negative bacteria. Yes, remove harmful immune complexes from the body. So the only options that don't fit in with the complement system functions are it repels infiltrating leukocytes and it protects the foreign objects or organism. Is this clear? Right, so that's the last question I had for you people for this assignment. If you have any doubts or want me to repeat any of these questions, I can do so right now. Or if there is any other topic that you would like me to discuss, we can do it right now or in the next session. But yeah, regarding assignments, that's all I had for you. Yeah, so we can stop this meeting here and I can meet you people next Tuesday with week 10 assignments. That's all. Thank you all for joining. Okay, I'll stop the meeting then. Rest of the people who are still here. Alright, let's meet next week, Tuesday at 6 o'clock. Thank you.